All right, lesson 1 in phylogeny and systematics. I want you to take a look at this tree of life here that represents a good number of prehistoric sharks. Sharks that we don't really find on Earth anymore today, but we can hypothesize that they existed based on various forms of evidence, fossils and remains of various types. Now, looking at this tree of life, I'd like you to note that the ones down here at the bottom are the most ancient. And then moving up the tree, we can see a progression. This tree of life can be called a phylogeny. So phylogenies are basically a kind of fancy technical term for a tree of life that shows a few different things. This can also be called a cladogram because it represents a clade or a group of classified organisms. So you can tell a whole lot about things from a tree of life. You can tell the supposed order or the predicted order in which they appeared in. You can associate them into groups that had a common ancestor that speciated or diverged. You can tell which ones are more closely related and which ones would be less closely related to one another based on how long ago they diverged, last time they had a common ancestor. There's a lot that you can tell about these. Now, what I would like you to do is take a look at this video and try to think about where it might go on this cladogram. A video here of a shark. Now this shark is a prehistoric shark, but they found one that was alive. Usually these things have only been caught in nets dead or washed up on shore dead, but this one was kind of swimming pretty much at the end of its life, possibly confused, and it was discovered off the coast of Japan. And if you look at this thing, it doesn't have a strong dorsal fin, it's very long, it had these frills on the side of it, and it indeed died a short time after it was discovered. Again, this is a prehistoric shark. It's basically a living fossil. It's one of the most rare organisms on the planet. It's almost never been seen, certainly not alive the way it was in that video. Here's another picture of that frill shark. Where would you put that based on its characteristics on this chart? You may notice that it doesn't have a dorsal fin. Some of these organisms do not have dorsal fins either. Based on its tail shape, where would you put it? Where do you think it belongs in this classification scheme here? Now, there is no right or wrong answer to this, but it's a question worth answering, because we would like to classify this organism with other organisms that it is closely related to. Now, there's an inherent value in doing that. If you know what groups exist, you can identify unknown organisms or newly discovered species. The fact that we have a category called beetles means that we can take new beetles and put them into that category, possibly into subgroups that represent it accurately. We can also use these identifications to establish evolutionary connections, relationships, such as the divergence of humans from other great apes a few million years ago. It also allows you to predict characteristics. You can predict that any newly discovered mammal will be warm-blooded, it will have hair, it will produce lactate that it gives to its offspring in the form of milk, and it will likely have a live birth. These predicted characteristics are representative of the group, and so certain blood proteins would be shared by groups, or other morphological or physiological traits. And then lastly, that allows you to treat other organisms that are in the same group similarly. For instance, if you do a test on a mouse and there's a drug that you're testing, if it works in the mouse, there's a good chance it's going to work in humans because humans are mammals as well. We share a lot of our physiology, even though we and mice appear to be very, very different. So these classifications and categorizations can be based on what's called biochemical evidence for a common ancestor. So the more close a structure is, and in this case the structure would be a chemical structure, or what we could call chemical homology, it indicates common ancestry. Things that are more similar would have diverged not so long ago. Things that are very different have had a long time to become different because they do not share an immediate ancestor evolutionarily. So we can use chemical homogeny to figure out relationships between species. One of the ways we can do this is by sequencing amino acids, figuring out what the sequence of amino acids would be in a given protein. A protein such as hemoglobin, carrying oxygen in your blood. Cytochrome C, which is a protein that is associated with the inner mitochondrial membrane that you have. And lastly, chlorophyll. So again, if we were to find that two proteins were relatively similar, then we can assume that their similarity is the result of a recent divergence on the tree of life. If we find that they are not similar in any way, then we can assume, or that they have many differences, then again we can assume that they diverged or became different a longer time ago, helping us to establish relationships and divergences. In addition, genetic analysis can help us with this as well. A lot of the same principles. We can look at the genes in our DNA. We can also look at the highly repetitive sequences that separate our genes and actually make up the large majority of your genome. These highly repetitive sequences accumulate mutations, but because those mutations are not ever made into a protein, because they're not part of a gene, those mutations are all silent. They don't actually affect the survival of the organism. And then lastly, mitochondrial DNA, which is inherited maternally from your mom. And so, again, by looking at the decoded genes of organisms in their genome, organisms that are very closely related probably didn't diverge on the tree of life very long ago. Humans and chimpanzees share something like 95% of our DNA. Any two humans share about 99.6% of our DNA. Whereas we and other mammals, such as a mouse and a human share about 81% of our DNA. So the further back you go on the tree of life, the more time there's been for our DNA to mutate independently of one another, and the less similarity there should be. One way that you can figure out how similar DNA is by doing this thing called DNA hybridization. So differences between two species DNA, say species A and species B, would be due to mutations, and mutations actually happen at a relatively predictable and certain rate. So mutations as a result of replication errors will occur every so often, but at a sort of reliable tick. And so any differences, say differences here or here, represented by the fact that these two DNA strands are not complementary to each other, means that there was a mutation at that point, at one point in time. More differences indicate that two species diverged a long time ago. Fewer differences indicate that they are more related and diverged not quite so long ago. So you take two DNA strands that are complementary to each other, maybe a sense and antisense strand from two species, and you see how many places they are still complementary, how many places they essentially have the same genome. In addition, you can take a look at a mutation rate like, say, 5 base pairs every 100 years. And then if you were to say, and I'm making these numbers up, but if you were to take two species that have 100 base pairs different in a certain gene, it wouldn't be that hard to figure out how long ago they diverged on the tree of life. You could actually make a prediction that could be supported later by fossil evidence. And so essentially what you need to do is take the 5 base pairs into account with the 100 base pairs of difference. Take 100 divided by 5, and find that it's 20. And that means that this 100-year cycle has happened 20 times. So 20 times 100, well, you get out your calculators and do it because I'm not going to for you. Chemistry.